Good evening, everybody. Welcome back to daily lectures by New Age Cosmetic White's Edtech Healthcare Consultancy. I am Dr. Bobby, and we're on the subject of molecular biotechnology. We're on the chapter of biomass utilization and bioremediation. And we're on the subtopic of engineering yeast transcription. And we were on to the last session where we were mentioning that this could be achieved using the above-mentioned approach of reprogramming of a microorganism by realizing of mutagenization or mutagenizing one of the proteins which is responsible for regulating global transcription of microorganisms. Now under the figure that shows the engineering of E. coli, we see that it's engineering of E. coli to produce isopropanol. A simplified overview of the biosynthetic pathway. The introduced portion of the pathway is highlighted in yellow. The enzymes involved in this pathway are as follows: several E. coli enzymes. Steps are required for the conversion of glucose to pyruvate. Pyruvate is converted to acetyl-CoA by E. coli enzymes. Acetyl-CoA acetyltransferase from a acetyl-butylicum catalase. And excuse me, the details have been shown here and the notes given here. And of course, we have B-acetyl-CoA acetyltransferase from acetyl-butylicum catalase, acetoacetyl-CoA transferase from E. coli catalyzing to production of acetate, and D-acetoacetate decarboxylase, from C-acetobutylicum catalyzes to synthesis of acetone, and secondary alcohol dehydrogenase from C-BIOTECH, which produces isopropanol from acetone. In order to engineer yeast to more efficiently produce alcohol, it is necessary that the yeast strain be able to tolerate high concentrations of both glucose and ethanol. To achieve this end, it is likely that the expression of a large number of yeast proteins must be altered. Moreover, it is by no means clear which proteins need to have their levels of expression either increased or decreased. Instead, one group of researchers undertook to reprogram a significant portion of yeast metabolism by generating a large number of randomly mutated yeast transcription factor genes. In a small number of cases, the modified transcription factor would alter yeast gene expression in a manner that increases the tolerance of yeast for higher levels of both glucose and ethanol. And in yeast, 15 different proteins bind to DNA and regulate the promoter specificity of RNA polymerase II to dramatically modify yeast metabolism. The gene encoding transcription factor SPT15 was altered by error-prone PCR. This amplification reaction produces a range of different mutagenized SPD15 genes. All of the mutagenized SPD15 genes were cloned in a plasmid vector and then introduced into wild-type yeast via S. cerevisiae cells. The transformed cells are grown in agar medium in the presence of 6% ethanol or 120 grams per liter of glucose. Any transformant that grew better on this medium than the native yeast was then more fully characterized. One particular mutant displayed a high level of cell viability and grew faster than the native yeast strain at ethanol concentrations of between 10 and 20 degrees centigrade. When this strain was examined in detail, it was found that SPD15 had 3 separate mutations, all of which were required for this activity. In addition, when the mutant was transcriptionally profiled using microarray technology, several hundred genes were found to be differentially regulated compared to the native strain, with the majority of genes being upregulated. Interestingly, a detailed analysis of the genes whose expression was significantly altered did not reveal that a particular pathway or genetic network was primarily responsible for the observed reprogramming. Finally, the yeast strain that was used in these experiments was a standard laboratory strain, so that to incorporate this approach into a commercial process it would be necessary to repeat the work with an industrial strain of yeast. If successful, this type of genetic manipulation could facilitate the development of strains of yeast that more efficiently convert glucose to ethanol. Utilization of cellulose. With an increasing world population and dramatic increase in the standard of living in different parts of the world, developing world, meeting the growing worldwide demand for energy, for eating, for transportation, and industry has become a major challenge for all countries for the 21st century and beyond. Moreover, in addition to being sustainable, the future energy supply needs to be non-polluting so that we may realize a reduction in greenhouse gas emissions. To this end, many countries around the world have begun to produce large amounts of alternative fuels in an effort to at least partially replace non-renewable fossil fuels such as oil and gas. In this regard, a major effort has been directed toward producing bioethanol. At the present time, Brazil produces large amounts of ethanol from the fermentation of sucrose derived from sugar cane, and the United States produces ethanol from cornstarch. However, the reduction of greenhouse gases that results from the use of sugar or starch-based ethanol is not as high as desired. And many socially conscious individuals have criticized this strategy of converting land from the production of food to the production of ethanol by energy. Moreover, sugar or starch-based ethanol is unlikely to provide more than a small fraction of what we require. In this regard, in 2008, the Chinese government announced that it would not follow any further increase in starch-based ethanol production because of competing uses as a fuel food. If the world is going to produce ethanol on a large enough scale to significantly lower our use, and of fossil fuels, so that ethanol will have to be produced from lignocellulosic waste products such as corn, stover, grasses, and wood chips. Thus, there is now, more than even before, a tremendous amount of both political and scientific activity directed toward trying to produce ethanol from lignocellulosic materials. Lignocellulosics. The polymers lignin, hemicellulose, and cellulose combine in various proportions to form a lignocellulosic structure, support system for nearly all terrestrial plants. This material constitutes a vast biomass that is often a waste product of agriculture, timber processing, and other human activity, and needs to be disposed of in a safe and efficient manner or used as a resource. It has been estimated that annually 10 raised to 11 tons of these polymers is synthesized in the biosphere with an energy content that is equivalent to around 640 billion tons of oil. Lignocellulose. Lignocellulosic materials, or cellulosics, have been grouped into three classes. Primary cellulosics include plants that are harvested specially for cellulosic content, structural use or feed value, example cotton, timber, and hay. Agricultural waste cellulosics are the plant materials that remain after harvesting and processing. Example straw, corn stovers, rice hulls, sugarcane bagasse, animal manures, and timber residues. Municipal waste cellulosics encompass waste paper and other discarded paper products. Components of lignocellulose. Lignin is a three-dimensional, globular, irregular, insoluble, high molecular weight, greater than 10,000 polymer made up of phenylpropane subunits with no chains of regular repeating units or any bonds that are easily hydrolyzed, either enzymatically or chemically. The lignin polymer molecule. There's many different types of chemical linkages between aromatic phenylpropane units. The physical and chemical characteristics of lignin are generally attributed to the last step in lignin biosynthesis, the non-enzymatic free radical-based joining of the phenylpropane units in a more or less random fashion in plants. Lignin is chemically bonded to the hemicellulose and wraps around fibers composed of cellulose. Lignin is responsible for the rigidity of plants and for the resistance to the mechanical stress and microbial attack. Hemicelluloses are short-chain heterogeneous polymers that contain hexose or 6-carbon sugars such as glucose, mannose, and galactose, and pentoses 5-carbon sugars such as xylose and arabinose. The three major types of hemicelluloses are xylans, which have a backbone of poly-beta-1,4-xylan, with side links to arabinose, glucuronic acid, and arabinoglucuronic acid, mannans, which are composed of glucomannans and galactomannans and arabinogalactans. The origin of the lignocellulosic material usually defines the nature of the hemicelluloses. For example, xylan hemicellulose is particularly common in hardwoods, and glucomanans are characterized of softwoods. Cellulose, which is the simplest of the components found in lignocellulosic material, is the most abundant polymer in the biosphere. It is composed of long chains of D-glucose molecules linked in the beta-1,4 configuration. Both cellulose and starch can be hydrolyzed to glucose, but their structures are very different. Starch is an energy storage molecule in which the glucose residues are linked in a manner that prevents a tightly ordered arrangement of the polymer chains. This open mesh-like structure is easily penetrated by water. As a result, starch is both water-soluble and readily hydrolyzable by amylases and glucoamylases. By contrast, cellulose is a plant-supporting structural molecule. The glucose chains in cellulose are arranged in a manner that permits them to pack together in a crystal-like structure that is impervious to water. Consequently, the cellulose polymer is both insoluble and resistant to hydrolysis. Nevertheless, cellulose is still a form of stored glucose, so it is the component of lignocellulosics that has the most potential for conversion into a variety of useful compounds such as alcohol. However, before cellulose can be utilized, it must be released from its complex with lignin and hemicellulose. For most lignocellulosic materials, this separation requires treatment with either a strong acid or a strong base, or the use of higher temperature and pressure. Regardless of how the cellulose is separated from the lignocellulose complex, the energy that is necessary to achieve this adds significantly to the cost of the final product. However, since the annual production of lignocellulosic materials is huge, effective ways of enzymatically degrading cellulose and hemicellulose are being sought. Chemical and enzymatic methods for the selective degradation of lignin are also being investigated with less success. Isolation of prokaryotic cellulase genes. A wide variety or range of bacteria and fungi are naturally capable of degrading cellulose through the concerted action of several enzymes that collectively are referred to as cellulase. Aerobic microorganisms typically new large amounts of these cellulase enzymes in the medium outside of the cell. On the other hand, in anaerobic microorganisms, cellulase activity is often found as part of a multi-protein complex, as called a cellulosome, that lies on the external surface of the cell. Cellulases consist of multiple copies of several enzymes with different enzymatic activities, including the following: endoglucanase, which hydrolyzes beta-1,4 linkages between adjacent glucose molecules within the amorphous, loosely packed regions of the cellulose polymer, thereby breaking the chain in the middle. Hexaglucanase, which degrades the nicked cellulose chains from their non-reducing ends and produces glucose. Cellobiose, 2 glucose units, and cellotriose, 3 glucose units. Cellobiohydrolase, which is often found in celluliting fungi, and is a type of hexaglucanase that removes units of 10 or more glucose residues from the non-reducing ends of the glucose molecule. Beta-glucosidase, or cellobiase, which converts cellobiose and cellotriose to glucose. The breakdown of glucose cellulose by microorganisms. If the bacteria or fungi that produce the various components of cellulase is slow and often incomplete, therefore genetic engineering strategies have been used in an attempt to create organisms with more effective cellulase activity. For this purpose, genes coding for the individual enzymatic functions of cellulase activity have been isolated from both prokaryotic and eukaryotic organisms. Prokaryotic endoglucanase genes have been cloned by the following simple yet effective identification technique. A clone bank of DNA from a cellular lytic Prokaryote is constructed in E. coli and the host cells grown overnight on solid medium containing a selective antibiotic. The colonies are then overlaid with the agar-containing carboxymethyl cellulose (CMC), a soluble derivative of cellulose, and the Apache plates are incubated at 37 degrees centigrade for several hours. During this time, the CMC molecules that are present in the immediate vicinity of a colony that both synthesizes and secretes an endoglucanase are partially digested. Transformants that synthesize but do not secrete that cloned endoglucanase are not able to degrade the substrate because it is too large to enter the cell. The digestive regions of the CMC are visualized by first flooding the Petri plate with a solution of dye, Congo-Red, which is not toxic to the bacteria, followed by a wash with a solution of sodium chloride. Congo-Red selectivity binds to a high molecular weight cellulose chain and gives a red color. Conversely, it binds weakly to low molecular weight polysaccharides and produces a yellow hue. The sodium chloride treatment stabilizes the binding of the dye. If a bacterial colony produces a secrete endoglucanase, it will be surrounded by a yellow halo. The background where the CMC is not being degraded will be red. This technique has been successfully used in the isolation of endoglucanase genes, Streptomyces, Clostridium, Anorobacter, Monospora, Erwinia, Pseudomonas, Salvibrio, Ruminococcus, Cellulomonas, Fibrobacter, and Bacillus species. There is no convenient plate assay for detecting cells with a cloned hexaglucanase gene, so immunological screening has been used to pick out the recombinant clones that express hexaglucanase, although this approach requires specific antibodies directed against the target protein. The protein does not have to be secreted to be detected. Recombinant cells can be lysed in situ, example by exposure to chloroform vapor before the cytoplasmic proteins are transferred to a nylon or nitrocellulose membrane for subsequent immunologic testing. In these tests, replica plates are used to ensure that viable cells are available for further propagation and use. Prokaryotic beta-glucosidase genes have been isolated by transforming a clone bank from a beta-glucosidase producing microorganism into E. coli and then selecting the transformants that can grow on minimal media with cellobios as a sole carbon source. Alternatively, clones that express beta-glucosidase activity can be detected with a chromogenic substrate such as a 5-bromo-4-chloro-3-indolyl beta-D-glucopyranoside BCIP in the plating medium with McConkey cellobias agar. In both cases, beta-glucosidase positive colonies turn red. Isolation of eukaryotic cellulase genes. The strategy of DNA hybridization or screening of either cDNA or genomic clone banks with a heterologous probe that has not been particularly effective for isolating cellulase genes because the sequences of cellulolytic enzymes from diverse sources are not very similar. To isolate the messenger RNAs, mRNAs that encode cellulolytic enzymes from fungi or plants, a novel protocol has to be implemented. A novel protocol has had to be implemented. Unfortunately, these mRNAs usually constitute only a small fraction of the total mRNA population. Therefore, it is often necessary to enrich for the target mRNA or cDNA to eliminate cDNA clones that do not carry the target sequences. To meet these ends, the technique of differential hybridization has been used for the isolation of a number of different induced eukaryotic cellulase genes as follows. mRNA is isolated from cells grown without cellulose, that is, non-induced cells, and from cells grown in the presence of cellulose to enhance synthesis of cellulase enzymes, that is, induced cells. Each mRNA population is fractionated in a sucrose gradient, and each fraction is translated into a cell-free system, either rapid reticulocytes or wheat germ. A cellulose-induced proteins is identified after separation of the cell-free translation products on a polyacrylamide gel by the presence of unique bands that appear from the induced cells. but not from the non-induced cells. This step indicates which mRNA fractions contain messengers that are induced by the addition of cellulose. The mRNA sucrose gradient fractions from cells that direct the synthesis of cellulose-induced proteins, and the comparable fractions from the non-induced cells are used separately to program the synthesis of cDNA. The cDNA sample from the induced cell population is cloned into a plasmid vector, introduced into E. coli, replica painted, and then separately screened with labeled cDNA from both the induced and non-induced fractions as hybridization probes. Clones that hybridize only with cDNA from the induced cells and not with cDNA from the non-induced cells potentially carry cellulose-induced genes and are characterized further. To establish conclusively which of the positive cDNA clones encode cellulose enzymes, the DNA from these clones is introduced into an E. coli expression vector, and after E. coli is transformed with these constructs, the protein products are detected with antibodies to enzymes of the cellulase complex. The sequence of each of the positive DNA clones is determined. In principle, this scheme can be used for the isolation of any induced eukaryotic genes. Manipulation of cellulase genes. There are a variety of uses for cloned cellulose genes. In some cases, the cellulose binding domain encoded by cellulase genes facilitates the purification of recombinant proteins. In other instances, the cellulolytic activity is expressed in organisms that can convert waste cellulose into a commercial product such as alcohol. Trichloroethylene. The compound trichloroethylene is widely used as a solvent and a degreasing agent, and as a result it is one of the most common contaminants of soil and groundwater. Trichloroethylene persists in the environment for years, is a likely carcinogen, and is regulated by the United States under the Safe Water Drinking Act to a maximum contaminant level of 5 parts per billion. Unfortunately, aerobic soil bacteria can reductively dehalogenate it to produce vinyl chloride, which is an even more toxic compound. Studies showed that some of the strains of P. putida that could degrade aromatic compounds such as toluene could also degrade trichloroethylene. Genetic study established that a complete metacleavage degradative pathway was not necessary to completely detoxify trichloroethylene. In fact, only the enzyme toluene dehydrogenase, which normally catalyzes the oxidation of toluene into cis-toluene dihydrodiol, was required. 4 genes are involved in the production of a functional toluene dioxygenase. These genes were isolated and expressed in E. coli under the control of the strong and inducible TAC promoter. When E. coli cells carrying these genes were induced by the addition of isopropyl 1-beta-D-thiogalactopyranoside (IPDG), an inducer of the TAC promoter, trichloroethylene was efficiently broken down to harmless compounds by the concerted enzymatic activities of the TART proteins encoded by these 4 genes. Although the initial rates of trichloroethylene degradation were lower than the E. coli than with the original P. putida strains, the E. coli cells maintain these rates of longer periods than P. putida. It has been speculated that the basis for these differences may be that E. coli membrane are not as susceptible to damage from trichloroethylene as P. putida membranes are. In variation of this experiment, a hybrid Pseudomonas strain with elements of 2 separate degradative pathways was constituted. Bacterial strains that can degrade the compound biphenyl use the enzyme biphenyl dioxygenase. Biphenyl dioxygenase is a multi-component enzyme encoded by 4 genes: BPH, A1, A2, A3, and A4. With BPH, A1 encodes a large subunit of terminal deoxygenase, an iron sulfur protein. BPH, A2 encodes a small abundant subunit of terminal dioxygenase. BPH, A3 encodes ferredoxin. BPA4 encodes ferredoxin reductase. BPH-A1, BPH-A2 are associated as a heterotramer. heterotetramer and catalyze the introduction of two oxygen atoms into the biphenyl ring. Ferredox and ferredoxin reductase act as an electron transfer system from reduced nicotinamide adenine dinucleotide NADH to reduce terminal dioxygenase. Biphenyl dioxygenase is quite similar in both structure and function to the enzyme toluene dioxygenase. Despite the similarities of the enzymes, biphenyl utilizing pseudomonads cannot grow on toluene, and toluene utilizing strains cannot grow on biphenyl. However, when the BPH-A1 gene encoding for the large subunit of biphenyl dioxygenase from PPU to the KF715 was replaced by homologous recombination with the TOT-C1 gene coding for the large subunit of the toluene dioxygenase from PPU to the F1, the resultant strain was able to degrade dichloroethylene. In fact, the engineered strain grew well on a range of aromatic compounds and also was very efficient at degrading trichloroethylene, demonstrating that it is possible to rationally engineer bacterial strains that can degrade a number of different compounds. The creation of degradative bacteria with novel biological activities was achieved by slightly different means by the creation of chimeric versions of the BPH-A1 gene. In this case, one BPH-A1 gene was from a strain of Pseudomonas pseudoalkali genes with the ability to degrade only a narrow range of polychlorinated biphenyl PCBs, while the other was from Burkholderia cepacia, which can degrade a very wide range of PCBs. Some of the hybrid genes encoded an enzyme with a wider degradative ability than either of the original enzymes, the native BPH-A1 gene and P's pseudo-alkali genes was replaced with hybrid genes by homologous recombination. It now remains to be seen whether these engineered bacterial strains degrade a range of PCBs on a large scale. Cell surface expressed enzymes. Currently, detoxification of organophosphate pesticides in the environment is performed by chemical treatment, incineration, or burial in landfill sites. Each of these approaches has serious environmental drawbacks. It would therefore be advantageous if bacteria that are able to degrade these compounds could be utilized in place of the methods that are currently used. Several soil bacteria, including Pseudomonas diminuta, Mg, and Flavobacterium spp, possess an enzyme organophosphorus hydrolase that catalyzes the hydrolysis of many of these pesticides, including methyl and ethyl parathion, paraoxon, chlorpyrifos dersban, coumaphos, cyanophos, and diazenon to environmentally innocuous compounds. Unfortunately, these bacteria, as well as E. coli engineered to express organophosphorus hydrolase, degrade these pesticides relatively because of the low rate of uptake into the bacterial cells. Thus, a novel approach was developed to solve this problem. E. coli cells were engineered to express organophosphorus hydrolase as part of a fusion protein that contained the E. coli lipoprotein signal peptide and the N-terminal portion of the lipoprotein and outer membrane protein A. When this fusion protein was synthesized, it was localized on the outer surface of the bacterium. This eliminated the problem of a low rate of pesticide uptake into the engineered bacterium. Cells with organophosphorus hydrolase on the outer surface had approximately 7 times higher activity than cells that expressed a similar amount of the enzyme intracellularly. Moreover, the enzyme activity of cells with organophosphorus hydrolase on the surface was extremely stable. Nearly 100% of the activity was retained after 1 month at 37 degrees centigrade. This concept is quite promising. However, to date, it has been tested only on a laboratory scale. Radioactive environments. The 26 countries worldwide that generate electricity from nuclear power plants, as well as those countries that have nuclear weapons programs, have generated thousands of radioactive waste sites. In the United States alone, there are more than 3,000 of these sites. It's been estimated that using BIOTECHNOLOGY, a cleanup would require about $200 billion. That's just a general know-how. Things could be much better right now. This is the 4th edition. Now, on to nitroaromatics. For many years, a large number of different nitroaromatic compounds have been used industrially as dyes, plasticizers, explosives, solvents, and pesticides. Many of these compounds are recalcitrant to break down, persist in the environment, and are now considered to be toxic and sometimes carcinogenic pollutants. For example, the compound 4-nitrophenol is formed by the hydrolysis of the insecticide parathion and is considered to be a priority environmental pollutant, in part because it leads to numerous human teeth health problems. Similarly, the compound 3-methyl-4-nitrophenol is a toxic breakdown product of the agricultural insecticide phenitrophenol. The bacterium Burkholderia sp. strain DNT facilitates the breakdown and detoxification 2,4-dinitrotoluene. First, the enzyme 2,4-dinitrotoluene dioxygenase removes one nitro group to form 4-methyl-5-nitrocalcol, which is converted to 2-hydroxy-5-methylquinone by the enzyme 4-methyl-5-nitrocatechol monooxygenase. Unfortunately, the 4-methyl-5-nitrocatechol monooxygenase has a very narrow substrate range, which includes 4-nitrocatechol as well as 4-methyl-5-nitrocatechol. However, this enzyme cannot efficiently use either of the seemingly similar substrates. In an effort to expand the substrate range of the enzyme, Burkholderia sp. The Burkholderia strain DNT 4-methyl-5-nitrocatechol monooxygenase gene was isolated and subjected to error-prone PCR and randomly mutated genes were closed into the plasmid vector. The library of mutated 4-methyl-5-nitrocatechol monooxygenases was transferred into E. coli cells by electroporation. And now on to the summary of bioremediation. This is the term that is applied to the use of microorganisms to clear the environment of contaminating substances. Many members of the bacterial genus Pseudomonas carry plasmids that encode enzymes capable of degrading aromatic and halogenated organic compounds. In most cases, a single plasmid carries the genes encoding enzymes for a specific degradative pathway. By combining plasmids from different pseudomonad strains within a single host, it is possible to create an organism with multiple degradation capabilities. In addition, by genetic manipulation, the range of substrates degraded by a particular enzyme pathway can be extended. Raw biological material is called biomass and is often used as a starting material in industrial processes. The use of milled grain for the production of alcohol or fructose requires Grain for the production of alcohol or fructose requires a number of the enzymatic steps. The enzymes that are used in these processes are often used only once and then discarded to enhance enzymatic conversions and decrease costs. Bacterial genes encoding enzymes that are thermostable, highly efficient catalytically, or tolerant of alcohol that have been cloned, characterized, and tested. To improve the commercial production of alcohol, some workers have genetically transformed the bacterium Z. mobilis with genes that allow it to utilize a broad range of compounds as carbon sources. Enzymes that degrade starch can also be used to facilitate the ability of microorganisms such as L. plantarum to ferment silage. Often as a sequence of processing biological material, large amounts of lignocellulose remain. This material generally has been treated as a waste product. However, there is no interest in using lignocellulose as a source for carbon-containing compounds, especially glucose, that can be used in other processes. Retrieving glucose from lignocellulose is not an easy matter. Lignocellulose is a complex of lignin, hemicellulose, and cellulose without harsh and expensive pre-treatment. It is refractory to enzymatic degradations. Recent research has focused on characterizing the mechanism of breakdown of cellulose to glucose. The genes for the endogluconases, exogluconases, and beta-glucosidases from a variety of organisms have been cloned and characterized, but to date there have been little success in formulating a set of enzymes that efficiently degrades cellulose in vitro on large scale. Now, on to the review questions. How would you genetically engineer bacterium to degrade trichloroethylene? 2. Outline a protocol that you would use to clone fungal cellulase genes. 3. Deactivate the role of alpha amylase and glucomylase in the industrial production of alcohol. How might genetic manipulation of the genes encoding these enzymes be used to improve this process? What is glucose isomerase? Why is it important? How and why would you modify the gene encoding this enzyme? Elaborate some of the advantages and disadvantages of using Z mobilis instead of Escherichia for alcohol production. How would you improve the industrial performance of Z mobilis? 6. How can Z mobilis be engineered to produce ethanol from xylose and arabinose? 7. Starting with Pseudomonas strain that can utilize phenol as its sole carbon source at 0 degrees centigrade. Pseudomonas strain that can degrade anthracene to catechol 35 degrees centigrade, and Pseudomonas strain that can degrade peat toluene to protocatechate 35 degrees centigrade. Suggest a strategy or developing a strain that can utilize phenoanthocene or peat toluene as its carbon source at 0 degrees centigrade. And number 8 will be explain how a Pseudomonas strain that carries plasmid PWWO and does not normally degrade 4-ethyl benzoate can be genetically manipulated to hydrolyze the compound. Suggest schemes for the isolation of prokaryote of endoglucanase and beta-glucosidase genes. What is a superbug? 11. How can L-plantarum be manipulated to improve its ability to ferment silage? 12. How can pesticide-degrading enzymes be expressed in the surface of a bacterium? How would you degrade organic environment pollutants in the presence of high level of radioactivity? And 14. How would you engineer yeast strains to effectively convert glucose into ethanol? How would you explain the substrate range of a strain of Bacteria sp that normally degrades 2,4-dinitoluene, number 15? And now 16. How would you engineer glucoamylase to be more efficient in degrading starch? 17. How would you engineer E. coli to produce isopropanol? 18. What is the design of cellulosome? How is it produced? And 19. How would you engineer E. coli to produce hydrogen gas from formic acid? So that's it about this chapter. The next chapter coming up would be plant growth promoting bacteria. We'll be back with you in a short while. Take notes, prepare well. Kindly do not commercialize any aspect of these lessons or the lectures. Best wishes as always. I'll see you soon in the next lecture. Bye.